My name is Jill Johnsen. I trained as an adult hematologist. I'm also expert in a lot of aspects of genetics, and I also do research. So I'm at the University of Washington in Seattle. And today I'm going to talk about the genetics of inhibitors. All right, so that's the preamble. These are my disclosures, and none of them are relevant to what we're talking about today. So first, I want to make sure nomenclature just means what are the words we're using, that you guys know how do we talk about changes in DNA. So DNA is the recipe book for our cells and how our bodies know what to build. And it's made up of 4 letters, A, T, G, and C. Each of those is a name for a molecule, but we can think of them as truly as letters, and those letters spell words that make up sentences and paragraphs and chapters of how our body builds itself. So we first have to talk about what kinds of misspellings do we have in DNA. So the one that we understand the best, because we've been able to detect it for many years, is something we called a single nucleotide variant, and that's where something's misspelled by 1 letter. So let's say the reference genome, which is the genome we all use as the normal, but I'd like to point out it's not normal, it's just the one we all use, so we all know where we are, it's the index. Let's say it has a T, and then we do sequencing on a person and they have an A. That A change, the change from a T to an A is what we call an SNV, a single nucleotide variant. It means we've changed 1 letter from the common one to a rare one. It doesn't necessarily mean it did anything. In fact, with most of these changes that in our DNA don't do anything. We're very tolerant of these kinds of changes. But every once in a while, if it happens in a gene where the misspelling changes what the word means, like in factor 8 or factor 9, sometimes it breaks the gene and makes it so it makes an abnormal. It reads the word abnormally, or it makes the word not make any sense at all. It's like nonsense. So we also have other ways that DNA can be variant between people, and one is called insertions and deletions. And that just means there's a little extra sequence relative to the common, or we're missing some sequence relative to the common. So sequence is inserted, there's some letters inserted, or we're missing some letters. Again, most of the time, it happens in a part of the DNA that isn't really part of an important sentence, word, or chapter. It might be the white space in the book. It might be the space between a chapter. But every once in a while, it can really mess up a word. And when we have extra letters in a word, it really makes the word hard to read. So if you think about how your brain can read a word, often we can get past 1 letter change. But it's hard when a piece is missing or there's a whole bunch of extra letters. It's a lot like that. So we call them indels for short. I'm probably not going to use that word very much, but in case it's on the slides, indels, that's what we mean. There's extra letters or missing letters. And these can be category of a larger set of changes that we call structural variants. So they're called structural variants because they, instead of affecting a very small number of letters, they affect a much bigger part of the DNA. So it affects the structure of the DNA. And that's really important because there are a number of structural variants that cause hemophilia A and hemophilia B. And one example of those is the factor 8 inversions. So you may have heard of a factor 8 inversion causing severe hemophilia A. About 45% of severe hemophilia A is caused by this. So basically what happens is, it's truly what it sounds like. Something's inverted, which means it's turned the wrong way. In this case, for a factor 8 inversion, there's a part of the sequence that looks very similar somewhere else, and that whole block of DNA has flipped around, and the result of that is it broke the factor 8 gene. So that's what those words mean. So the main take home here is there's lots of different ways that DNA can be different between people, and we call that variation because most of the time it doesn't mean anything, but every once in a while it causes a problem. So, all right. I know we don't need this, but this is orienting us that we're talking about hemophilia A and B right now. And if someone wants to talk about inhibitors in another disorder, I'm happy to do that. I just made the content for hemophilia in A and B, but it absolutely happens in other disorders. So both hemophilia A and B are what we call X-linked disorders. That's because the genes that encode factor 8 and factor 9 are on the X chromosome, which is one of our sex chromosomes. We have an X chromosome and we have a Y chromosome, and we're going to talk more about that. So hemophilia genes are on the X chromosome. We know that the main complication of hemophilia is bleeding. And in males, which is the predominant pattern that's talked about in the textbooks, it's joint bleeds, soft tissue bleeding, injuries, surgeries. And I wanted to point out that females also can have those kinds of bleeding. When you look at females, their most common bleeding is heavy menstrual bleeding. So the point is, people who have hemophilia have bleeding. I think that's not news. And but I wanted to point that out because there's a lot of textbooks that say the main complication is inhibitors, which is very, very important. But really the main complication is bleeding, and then we're going to talk about inhibitors. The other thing is, how do we define hemophilia? Clinically, severity is defined by the factor activity level, not how much or how often you're bleeding. And when we use words severe, moderate, mild, it's my opinion we're not doing anyone any service, because we're really not conveying how much are they bleeding. What we're conveying is what are their factor levels in the lab. So they're very important, and they correlate with overall how much people bleed and whether or not they need prophylaxis and their risk for inhibitors. But it doesn't tell you in any one person how much are they bleeding and what's their risk for inhibitors. I got this from an open source graphic that you all can also access. So if because it's open access, so it's available online, and the references here. And basically, it's what's called a graphical abstract, which means that it's supposed to use pictures to teach an important concept. And it's geared towards the science community, but I like these pictures for everybody because I think all of us are scientists, and we're all in this boat together. So I'm using these to teach the key concepts. So I also want to point out that much of what I'm going to present today is in hemophilia A, and a lot of the principles apply to hemophilia B, but we know it's not the same. But I don't have enough data to show you. So one clue just that hemophilia A and B inhibitors are not the same is that hemophilia B inhibitors come along with allergic reactions and hemophilia A doesn't. So clearly our bodies are telling us they're not the same, but I just don't have enough information to tell you why. So I wanted to make it clear a lot of what we're going to talk about is using a literature, graphical teaching points from A, and that we often infer that to B, and that sometimes that's going to be right, and occasionally that's not going to be right, and I can't yet tell you when it's not right. All right, so treatment of hemophilia, the mainstay of treatment is to provide factor replacement. So sure, there's a lot of non-factor prophylactic options. When someone's bleeding though, factor replacement is still the mainstay of where we reach. And so that's where we start talking about inhibitors. So our body's immune systems are designed to recognize foreign invaders and fight them off. And the way that it knows it's foreign is it first has to know what is self. So our immune system is constantly teaching itself who's friendly and who's foe. Because if you start attacking friendly, you're going to have an autoimmune disorder. We don't want to do that. We need to not attack ourselves. In hemophilia A and B, remember we're low or absent in a factor level. And that factor level means that we might not be teaching self for that factor. So when we see that factor in some people and not all, which is super interesting that it's not all. When you get exposed to factor, your immune system sees foreign invader and does what it's supposed to do, which is to fight that invader off. And when we fight off an invader, we want our immune systems to remember we just saw a bad guy. And so we make memory so that the next time we see it, that we know to call in the troops right away with a brisk response. I've learned that's an enemy. It's flu, it's COVID, whatever. It's that nasty factor level I just saw. And that's what makes us able to fight off infections and live a life where most of the time we're not sick. But our immune system, when it wasn't able to teach self, means it's recognizing something we really don't want it to recognize, because that was also a very effective treatment. So that's the concept of how do inhibitors happen. It's because of the gene changes that gave us hemophilia, we also can't recognize the normal factor as self. Making sense? All right. So there's a lot of pieces to that that are specific to very different kinds of inhibitor responses, but all of them have that same principle. All right, so this is the definition of inhibitors, and how we find them is that factor doesn't live very long in circulation, and usually there's an antibody that interferes in our assays. That means that the factor level activity is neutralized in our assay. And so that's called a Bethesda unit because that's the assay we're all using. And I'm not going to talk much more about how we measure it, other than to say that the presence of that definition is underlying all the data I'm going to talk about, because we have to define what an inhibitor is in order to study it. And we know there's a lot of risk factors for inhibitor development, and we also know we absolutely don't know them all, and we don't know which ones are really important in which context, and this is the reason we need to do more research. But what we do know is which misspelling in the gene, what kind of misspelling in the gene that caused hemophilia A or B is definitely a risk factor for inhibitor development. Also, something's running in families. There are some families where multiple people will get an inhibitor and others that will not. And we've seen that enough time that we can do statistics and say family history is definitely a risk. So that could be the genotype. They've all inherited the same gene change, but it looks like it's probably more than that. So we think there are other genes involved and maybe other environmental things that they share that are going on to put inhibitor. Also, what factor VIII or factor IX product was used, what we're inferring from factor VIII, how often it was seen, whether or not it was seen as part of a routine prophylaxis or treatment in the setting of fighting off, let's say, a joint bleed or an injury. That's a very different immune state. So if our immune system's already up and running because we have a tissue damage, maybe we're more likely to see that factor VIII as foreign. Whereas if we see it when everything's very calm, maybe our immune system is more inclined to think, oh, maybe it's self. And that's a really important teaching point for our immune system. Another piece that you guys need to understand as we go into the genetics is the immune system is also encoded by genes. And there's a lot of differences between us in the genes that code our immune system. And that's because we want, as a population, to be able to fight stuff off. We need to have diversity in our immune responses if human beings get to keep being human beings over generations. So there's lots of variation in those things. I don't think we're going to need to go into tons of immune concepts, but there's a couple that are important to know. And one is we have a first immune response. And as part of that first immune response, our immune cells take the factor and they kind of gobble it up into pieces and they present it to our immune system and they're like, are any of these very scary? And if some of them are very scary, they'll engage with T cells and B cells to make our immune response. And one of the ways that they gobble all that up and put those little proteins out in sort of an immune buffet is something called the HLA system, also called the MHC system, which is a bunch of letters that aren't helpful other than to say they're really, really different between people. And those differences are implicated in differences in immune responses in lots of disorders, including like autoimmune disorders and other things. So they're plausible to also be a difference that's important when we make an inhibitor, because if you can't present that piece of the factor to your immune system at all, then it's never going to see it. So there's going to be some HLAs that are protective. So that's why HLA is important. We're going to talk about that a little bit more in the genetics later. There we go. And then the other thing is the timing of everything, because in order to gather the data to understand what's going on with the immune system, we also have to understand the timing of what's our immune system doing when we see factor, and then what does it do when it's not seeing factor? Because it's important when we ask a question, is someone's immune system able to see factor? So when the first response, which is where the immune system didn't know that it was about to see someone it thought it was foe, that's the time where it's starting to make memory. But it needs to see it a few times before it's really like, oh yeah, bad guy. And then once it knows it's a bad guy, the memory is in there through repeated exposures, each time it's going to have a bigger and bigger response. And then if it stops seeing factor for a while, it starts to sort of lose its memory, not completely, but then that does mean like at some point, like if you go for a period of time without any factor, the antibody levels can go down, the inhibitor levels can go down. So years later, maybe the inhibitor levels really low, you might get away with a few doses of factor while your immune system is remembering what it thought, 10 years ago. So those are all things that are relevant to how are we assessing how someone had an inhibitor response. And then the last thing is, well, there's also some genetic components we think in how people respond to the treatments that we give once an inhibitor has developed. So acknowledging that there's other treatments and bypassing agents and antibodies. Also to remind us that immune tolerance induction has been a mainstay of trying to get that immune system to just be quiet. And so what the idea of immune tolerance is that you just overwhelm the immune system over and over in whatever context to trick it into thinking, oh, I made a mistake, it's actually self. You're trying to get it back to thinking it's self. And some people can be tolerized that way, but some can't. And I'm going to show you a little bit of data from that we think there's a genetic component to that as well. All right, so let's talk about the genetics of hemophilia. So I've already told you, and you already knew, I'm telling you guys mostly what you know, I think is true, is that the genes are in the X chromosome. So that becomes really important when we talk about things like how is it inherited, and also what is self and not self. So, so first of all, it's on the X chromosome. And I want to point out that although a positive family history for hemophilia is informative, a negative family history does not mean that a person doesn't have hemophilia, because we can have new misspellings happen all the time. There can be new variants, and this is true for factor VIII and factor IX. And then, or maybe there's just a small number of people in the family, and we just don't have enough people to have realized, there haven't been enough people that had a chance to inherit it. We also underestimate or can misdiagnose hemophilia, particularly if it's in a female or if it's in a non-severe male. Sometimes it doesn't realize that they're bleeding, especially if there's not another family. They don't realize it's hemophilia for a long time. So, and then, we have people that are adopted or live very far from their families as young children and just didn't know there was a family history. So absence of family history does not deter me from considering the diagnosis, but a positive family history is super helpful to point us towards the right diagnosis. So let's talk about it. So this is just a little cartoon. We can't see the mouse. This is good news. Okay, so in a person who is XY, which is the most common combination of sex chromosomes for a male, they have one X and one Y. So if their X has the misspelling that causes hemophilia, and in this cartoon, it's that darkish blue color, then every male who has that combination will have hemophilia. And the reason is there's no other normal X around that might be able to produce a normal factor level. So all XY males who have that misspelling all have hemophilia. In a female, and the most common sex chromosome combination in females is XX. Usually what you have is one copy of the X chromosome with the misspelling that causes hemophilia and one copy of a normal factor gene. And in most, but not all females, that other copy is making some amount of normal factor VIII. For that reason, that female has a much better chance to think that factor VIII is self, because she's making it from a normal chromosome, if that makes sense. So inhibitors are much less common, but not zero. There are definitely females with inhibitors, and we need to remember that that can happen. So then when we think about inheritance, this male, let's say they partner with a two normal X chromosome female, every female offspring, daughter, will get his abnormal X. So that's what we call obligate carrier, which is a word I don't love, but that's what we call it. And that means, so every daughter of a male with hemophilia inherits that gene change. So that's an important concept. And remember, every female who inherits that gene change is at pretty high risk to have bleeding or a diagnosis of hemophilia. None of the XY sons can inherit the gene change because their X must come from the mom. All right. Then what happens when we have a female who has one affected X and one unaffected X? Still half of the X's on average, rolling the dice every time. So for those kids, one of the two boys on average will inherit that. Sorry, this one of the two boys. And remember, we learned if you're XY, then you just have hemophilia. There's no other normal gene. And then one of the two daughters will inherit that gene change and be at risk. Does this make sense so far? I see nodding, we'll go on. Sorry, I'm not good at advancing the slides. There we go. So one of the things that we have a fair bit of information about is what we call the genotype or the gene change that happens in hemophilia. And that's because we do a lot of genetic testing clinically and also as part of our research. And the reasons we do those is because it's helpful in a diagnosis. It's helpful in establishing some aspects of management. Sometimes it helps us predict the likelihood of inhibitors. It helps us do diagnosis and counseling in the family. And it's helpful for prenatal and neonatal testing and care. So that's why we have a lot of it. And we're going to talk about the inhibitor aspects today. But as some of you know, I will talk your ears off about genetic testing. So if you want to talk about that in another setting, I'm happy to do it. So this is some data from the My Life, Our Future genotyping program, which I was privileged to be part of. And this just the main take home from this slide on the left, or whichever side that is, your right, is a list of all these different types of DNA changes. Remember back at the beginning, I was telling you there's all these different ways that you can misspell. And then what you can see here is by disease severity, which is the factor level. And if it's hemophilia A or B, how common was it to find that DNA change by which disease severity? So you can see that severe hemophilia A and B look different than non-severe hemophilia A and B. But importantly, severe hemophilia A and B can still be kind of any of the possibilities as well. So there's a difference in the proportions, but all kinds of variation can cause hemophilia. So within there, in people who have severe hemophilia, and this almost all comes from hemophilia A, we think that there's probably a difference in the ways the gene change matters, because we think some gene changes just completely break the gene. It can't make anything. It can't make RNA. It can't make a dysfunctional protein. And then we also think that there are some what we call non-null, which means that they're not making enough factor to help hemostasis. Like it's all less than 1% factor level, but it might be making a little bit of protein. And we think for inhibitors that might matter, because if you make a little bit of protein, your immune system might have a chance to see it and think it's self. That makes sense. So some of the genotype change we think we're seeing in severe hemophilia is probably this. It is very hard for us to right now classify with high accuracy a null versus a non-null variant. But overall, if it's one of these big gene changes I told you about, where we're changing big blocks, we think that's worse. And if it's a little tiny change, maybe there's a chance that nature has a way to read through that and make a little bit of peptide. So that's the main difference between the list on the left and the list on the right is big gene breaks, little gene changes. But overall, that doesn't help us with a single person who's in front of us. And is the point. Like we're supposed to get to the point where we can help individuals in front of us. So there was a pretty seminal meta-analysis, which means that the researchers took all the literature that was available to date and reanalyzed it as part of one big analysis to get to enough people to draw conclusions. And they said, yeah, genotype does matter in inhibitor development. And this was published in 2012. And that really just established what we've been suspecting for a while, that the genotype really matters. And then this is also saying big changes worse than little changes. And I'm going to present now some data from the My Life, Our Future program to talk about what have we been able to learn from this program. So I'm hoping some of you may have participated in the program. So this was a US national clinical genotyping service, basically offered at HTCs throughout the United States between 2013 and 2017. And over 11,000 people participated. It was a partnership. Barb Conkle was the PI. I was in the back room in the lab at that point. I'm now in the front room. But so that was my job, was to be in the back. And it was a partnership of Athen and NHF. And then it was funded by Biogen and then Bioverativ. And it was a massive partnership with the HTCs. We had 107 HTCs by the end. So it was a huge accomplishment to do a genotyping program like that. And we returned genotypes to over 11,000 people. Those people were also offered the opportunity to participate in research, and over 80% of people said yes. So those yeses make most of what I'm about to show you next possible. So thank you to everyone who participates in research. So we do have a research repository that I'm now privileged to be the lead of, a new partnership. And so the plan is that we keep on going to learn new things. So first, what did we learn in the program? So this is again, the slides will be available to you guys and they're also published, but I'm just highlighting the main points. Was that in hemophilia A, we broke people down by if they have severe hemophilia A or basically non-severe hemophilia A, based upon, our hypothesis that if you make a little A, it's different, it's different than if you don't. And so what we found is a very high rate of inhibitors. If you had something that we hadn't classified before. So we have a new class of genotype I'm going to show you a cartoon of in a minute, which is what we call a complex intron 22 inversion. So that was one of the discoveries of the program, is that's a very high rate of inhibitor. And then also if you have a big chunk of the gene deleted, high risk of inhibitor, like we're talking 50% risk. Also, a finding was that if you had severe hemophilia and we couldn't find the genotype on clinical testing, you were also in the highest risk category. And that's probably because there's a big change that's happening that our genotyping assay can't see. And through research, I can tell you so far, everyone we've been looking for, that is the case. So that we have some people very high risk, 50% of people with that genotype. And then we have sort of a next category where it runs to like a quarter to a third of people. And that's the inversions and then and sort of misspellings that we think break the gene quite a bit. And then we have some others who they have severe hemophilia, but have lower rates of inhibitors, like with missense, more like 11%. And missense is like where you change one amino acid to another amino acid, a smaller change. And then importantly, also in non-severe hemophilia, we also see about an 8% rate of inhibitors with missense. So although rates are much different between severe hemophilia A and non-severe, what importantly it is, and it's not zero, we have lots of people who still will make an inhibitor with non-severe hemophilia, and also it's almost always a missense that does it. And there are some missense that are higher risk than others. So here, this is thanks to My Life, Our Future, enrolling a nice proportion of people with hemophilia B. This is the first real kind of decent scale look we've had a chance to see in hemophilia B. And we see a similar theme, but the numbers are smaller, so the statistics are a little harder. So we're just sort of describing to you that if you're missing a chunk of the gene deletion, again, around 50, a little more than 50%, that's really similar to hemophilia A for that same classification. And then we have what we call stop gain and these frame shifts. Those are things that tend to break genes a lot, but on average in severe hemophilia, 12% risk of inhibitors. And then we do see a small risk, but also associated with missense at 2% and not severe. So it's a similar trend, but again, I'm nearly certain this is a different immune beast. And so the clue is here that genotype is important, but there's probably something else going on with the immune system. How are we doing for time?
13 minutes.
All right, excellent. So I just want to show you what it means to have a complex inversion, and this is where it matters what's in your genotyping report. So this is where the genes flipped around. It just means that we also found another change with it, and almost always it was a deletion or an insertion. So, which is probably the thing that's causing that high inhibitor rate. So complete genotypes are important. Almost all genotyping labs do not go looking to see if you have a complex inversion. They identify the inversion and stop. Another thing we were able to look at in My Life or Future, there had been a conflicting literature about if someone identifies as Black or Asian or Hispanic, that those seem to be higher risk categories, but it wasn't always true in all the studies. And so in My Life or Future, we can say, yeah, this is a significant risk. So if you identify as Black or African American in the study, it's 30% instead of 20%, which is a big difference and statistically significant compared to white. I highlighted in blue what I find, it's not significant because there weren't enough people, but quantitatively, those are high rates to me and people who were in the study who identified as Asian, Native Hawaiian, or other Pacific Islander. And this is the first study, because we need a sizable study to see this. This is the first time we had a clue of that signal, but that needs to be replicated in other studies to know if it's true. Because what if they were just all related? We also learned that families are risk. And then it's a small difference, not Hispanic versus Hispanic, but it's statistically significant. And we were able to analyze it independent of race. So as a standalone risk factor, being Hispanic, Latino, Latina, Spanish origin, identifying is a higher risk for inhibitor alone. And we were also able to do this in hemophilia B, which I believe is the first time. And we saw even more so. Self-identifying as Black, as African American, much higher risk for inhibitor. And we also see even a clearer signal for Hispanic or Spanish origin. Dissecting whether or not this is a genetic risk factor or some other related socioeconomic determinant of health, we can't dissect it out. But one researcher in our research project tried. So this is a separate analysis of people who said yes to research. This is a very busy chart that you don't need to remember, but it'll be in the decks. But basically, it was looking to confirm that the signals that they expected to see from the larger study were present in this research data set, and they are. And then what they importantly did was they went and looked, and I'm really glad they did it. There are gene changes, misspellings in the factor gene that are much more common in people who self-identify as Black than white. And the thought was, well, maybe that's a difference between self and not self. Like maybe because we've made all our drugs from the white version. So if I give the white version of 8 to someone who has that other version, maybe it's because of that. So these are what we call haplotypes. It's the combinations of letters that are most common. And it turned out that it wasn't associated with risk of inhibitors. That was a really important thing for someone to do, because if it had been yes, why don't we just engineer a different version of factor based upon what your sequence is? So it would have been really cool to understand that that was part of it, but we now know that that's not part of the story. In a different research group using My Life or Future, looked genome-wide and just said, just ignoring everything else, who's at risk for inhibitors? What other DNA changes? And what you can see here, this is what's called a Manhattan plot. And the colored dots are basically regions where there's a gene change where there's a statistically signal that something is associated with inhibitors, and it's right over HLA. Remember, HLA is the part on white cells that says, here's some factor VIII or not. So this was good because this one makes sense to us. And this is just the table saying there were several different versions of HLA, that if you had that, you were at higher risk to have made an inhibitor. And that same study also identified a few other genes that looked like they were implicated in whether or not you made an inhibitor. So this is fantastic clues for us to go bird dog. What are the other things in our genome that might be putting us at risk? Because what we really want to do is say, as for the person in front of us in clinic, as a provider, I want to know what is your real risk. And it's not satisfying to say 30%, because you're one person, you will be all or nothing. And also, if we can figure this out, maybe we can find interventions that prevent or treat inhibitors better. And then I also want to show when a different researcher has gone and trying to use new AI tools and machine learning using the My Life or Future data set to say, what else can we learn from these patterns that might be associated with inhibitors? And so they find they found these DRBs are HLAs, the mutation type. And so they do find some of these patterns that we expect them to see. So this, I think, is a very encouraging path forward, where our conventional statistics might not find something that these new methods might be able to find something. And then lastly, I want to just talk about the genetics of ITI. So one of the challenges in research is you have to see events enough times to be confident you're not seeing them by accident. So this is where we're now in a very small group of people, which is people who went and developed an inhibitor of a certain type, and now they've agreed to do research in ITI. So thanks to the parents of these kids. So over 100 kids. And one of the things we learned is the genotype also is important in who responds to ITI. That's the take home. So there's genetic components to that too. And you can see here that the high risk group, they both had worse responses to ITI and less durable responses. And then I also want to talk a little bit about, this is very brief, and the IRB says I have to do it completely, so I couldn't just select a slide. So I'm going to very briefly talk to you about the Hemophilia Research Program, which please come talk to us if you're interested, and I'll give you a QR code then. But basically, we're looking to, one of the main purposes of this is relevant to this. So I have to show you this slide, but you already know what hemophilia A is. And this is hemophilia in pregnancy, and I want to emphasize that moms who have a gene change are at high risk for postpartum hemorrhage, regardless of their factor level. This is immune inhibitor formation, and again, this is kind of making the main points we've already made. And the study, one of the main points of this study is to understand the factors that drive factor VIII immune response. So the way this study works to both understand bleeding risk in pregnancy and inhibitor formation in childhood is we're starting with a pregnant mom, and we're following mother-baby pairs from before birth through several years of life. And we're looking at all these genetic things and other factors to better understand mom's bleeding and baby's inhibitor risks and development. So I'll give you this slide that's a QR code to the study if you're interested. And my next slide is thanking you all for your attention so that we have a minute to talk. And it's a real privilege to be invited to this event. So thank you for letting me come into your spaces.
For or find any relationship to hemophilia X that did not get hemophilia through hereditary and versus a genetic mutation.
Okay.
In the development of an inhibitor.
So, so the question is, in non-hereditary low factor VIII, which I think you're talking about acquired hemophilia would be right, where they've made an immune response to factor VIII. Oh, okay. So how do we get to? Oh, a new mutation in the family. Oh, okay. Got it. Yeah. So it's still hemophilia, still caused by gene change. So, no, that's a really good question. We have not. The main studies that have looked at that difference are, to my knowledge, have really been looking at the question about baby's brain bleeds after birth and the difference of knowing and making a birth plan because you knew that baby was coming versus one that didn't. So it's a really good point. I don't think so, because most of our studies have started with known kids right at the time they get their first factor, and most what we call de novos are found out when they bleed and get treated and then get worked up. But I think that's a great question someone should ask. To my knowledge, no. Yeah.
So if you have a known genotype, so my son's a G stop. So he basically will never tolerize with your notes, right?
So, if I can't give it individual, so I'm gonna like generalize the question, which is like, okay, so we have what we call a gain of stop, which means that the protein sentence stops prematurely before it gets to the end. For almost everyone, that means there's no peptide. There are a couple tricks of nature that read through those and make a very, very low level of abnormal peptide. So for any one person, I don't know, but as a class, we think that those are more prone to make inhibitors and would be more prone to have a hard time tolerizing overall. Yeah, and which I think is where we really need to understand what's the immune process of tolerization too, so that we can if I could take a prediction and say, here's a really high risk genotype, and we figure out something about inhibitor immune system, maybe those first factor VIII exposures happen with something else that tries to turn everything down. Yeah.
When we get to the point where gene therapy might be available for the inhibitor community, do you think the genotype will make a difference in whether you'll be successful?
I think so, but I don't know. There's so much we don't know about continuous lysing factor versus the version that created, that we know so much about with inhibitors, which was usually a little kiddo who got kind of intensive factor treatment. And then in a short period of that time of life where the immune system's different than an older child or an adult, that's where most of the cartoons we draw about what happens with the immune system have been based on. So there's a lot I don't know. Oh, there's so much I don't know. But one of the things I don't know is what's the difference between that kid from the teens and a kid now who's on ME or some other non-factor treatment, and they're only getting their 8 every once in a while? Does that turn into what we saw in non-severe? Like maybe some of it was the schedule. I don't know. And so maybe, but then again, now they're probably just getting it because they bled as opposed to prophy when they're not bleeding, which is a different immune context. So the same question I would have would be, I don't know what it means to have it around all the time versus this. I have to think that's different. There are other reasons to think that it might be tolerizing, but it might also make it so that we attack the cells that are making that factor, and that would be bad. So no, we're not going to know without research. I think, so that's like in an existing inhibitor. Then what do we do when we start making long-lasting or potentially curative therapies through gene modification available before they've had a chance to tell us if they make an inhibitor? This is another piece, right? They haven't had that many exposures. Does an exposure day on a potentially curative therapy count the same as this intermittent stuff? I bet it isn't, but I don't know which way. Maybe it's better, it might be worse. So we just don't know, but we have to answer that question, because what's unacceptable is to say we have a whole class of people that don't have access to long-lived or potentially curative treatments. Like that's just an unacceptable. Yeah.
So I come from a generation prior to blood clots.
The 40s, the 30s. I mean, we had cryo for a while.
But I mean, that's the first one that I ever used, besides mobile. But I'm talking like clotting.
So you're talking about concentrates and recombinant versus.
But, all right, and we've always heard the term inhibitor development.
Yes.
And my doctor, who diagnosed me in 1969 at 2 years old, I asked her about my inhibitor. And I was 5 when I was diagnosed, but products did not, cryo didn't work even up until then. And she said that I was probably born with it. Are you finding that situation in your study, that you're born with an inhibitor instead of developing an inhibitor?
Yeah, so to our knowledge, that isn't a thing that happens, but it is part of the reason we're asking the questions in that heart mother baby study the way we are. I'm not aware of a baby where we had access to cord blood or first day of life samples that came out with a true inhibitor. But the question is, did they come out primed to make that? Because we remember in the life cycle, we have that first immune response, and then we have the later ones. So a hypothesis that's never been tested, but we're asking now is, are there some babies that come out already primed? And my crazy hypothesis, maybe mom's factor VIII was their first 8 exposure and not the drug. And that might matter what they've seen at that point. And the other thing is there's a weird phenomenon in the general population where some people make antibodies to 8 that are not inhibitors. What if mom, and placentas are very good at taking antibodies across placentas. What if whether or not mom has one of those matters? Because that's another thing that targets a molecule to say, immune system, pay attention to this. It's sort of like, they shot the little arrow and are like, immune system, pay attention to this. So we have a lot of things we just don't know. But certainly there are some kiddos who it's not 20 exposures, it's like 4 and 5. And that's a difference we don't understand the reason for. The genotype does play a role in that. It looks like the very bad genotypes are probably also the earlier responders, the higher titer responders. So have we seen a kid come out with an inhibitor? Not to my knowledge. But we're missing all that data around birth. Like nobody's really looked systematically then. Most people are looking months later when we're doing first factors, or there's a brain bleed and they get their thing, but they don't have an inhibitor till later. It's a really good question. It seems like if mom had severe A and had an inhibitor, would that teach baby's immune system? Like we haven't had a chance to study that. But does that answer your question?
Sure.
I'm happy to talk more later.